We left off here, and we were going to start talking about fertilization. So I told you that this particular structure right here, this thick goo that goes all the way around an egg, that was going to be important to our story. But before we talk about fertilization itself, let's review the histology, the microscopic anatomy of an individual sperm. Sperm are very modified cells. They get made in huge numbers. They're made rather sloppily, so a lot of them aren't very functional cells. But one of the things that's difficult about being a sperm is that in order to be real hydrodynamic, in order to be able to swim really well, they can't have like a big bobblehead. So they needed to have their nucleus really shrunk down. So a normal nucleus of a cell might be like that big, right? And this has been really shrunk down. And it always reminds me of, you see those packages you can buy and you can stuff it full of blankets and linens and pillows and then attach a vacuum cleaner. It pulls all the air out of it. It makes it like this big. That's kind of the way the nucleus is in a human sperm cell. The nucleus sits in this region called the head of the sperm. And also in the head of the sperm is this guy, the acrosomal vesicle, very important to our story of fertilization. Then we've got a midpiece, and in the midpiece, that is where you'll find lots of the equipment. That's kind of like the engine that makes the tail able to swim. Also in the midpiece are mitochondria. And mitochondria that are inside of your cells, all of them came from your mother. None of them came from your dad. So if I look inside of your cells and I look at the genetic material that's in your mitochondria, they are 100% moms, 0% dads. And your dads, by the way, they all came from his mom too. That came from her mom, etc. So mitochondria. So now, the important part of the sperm for this part of the story is the acrosome. The acrosome is filled with digestive enzymes. Now, when I said digestive enzymes, you must have thought to yourself, what? Like, why would there be digestive enzymes in a sperm? What does that have to do with it? Well, when we use the term digestive enzymes, we're always talking about enzymes that will take big molecules and break them apart. That's what a digestive enzyme is. So yes, the digestive system uses digestive enzymes, but so does the acrosomal vesicle. The acrosome of the sperm is going to release digestive enzymes that will allow a sperm to kind of eat a tunnel through this stuff, the zona pellucida, so that that sperm gets the opportunity to fertilize that egg. A fertilization event will only happen if the cell membrane of the sperm actually touches the cell membrane of the egg. Okay, good. So let's talk some more about fertilization. Okay, so fertilization happens when the genetic material of the sperm and egg combine. So the DNA of the sperm, the DNA of the egg, and that won't happen until the two cell membranes touch. And when a sperm gets to an egg, it bonk, bonk, bonk is going to bump against the zona pellucida. Now, in movies and stuff, they always show all of the sperm racing to be the first one that gets to the egg, and that the first one that gets there wins, but that's not true. As a matter of fact, the first one that gets to the egg almost never is the sperm that fertilizes the egg. And that's because of this issue, capacitation. Sperm really are rather fast. And when the sperm are deposited here in the vagina by an ejaculation, then the sperm need to race all the way up here to the infundibulum of the uterine tube. A couple of things about that. One thing is sperm are not particularly bright, so they don't know where they need to go. How do they find their way at all? And the truth is, when the uterus is ready for ovulation, it's not just conditioning the endometrial lining, it is also creating a layer of mucus that is going to be kind of like a yellow brick road, like from The Wizard of Oz, yellow brick road for the sperm. So a perfect quality of mucus, one that's very swimmable and has got markers in it, will guide the sperm so that they know where to go to find the egg that they're going to fertilize. And it's true that they can get there actually pretty fast. It only takes them about 10 minutes to get up to the infundibulum. Is there an egg there? Maybe there is, maybe there's not. Sperm can stay in a woman's reproductive tract just like milling about, ready to find an ovum for about 48 hours. In some people, it can be up to 7 days, and that really depends on the immune system of the woman's reproductive tract. But remember, the oocyte only lasts for 24 hours. So they get up there really fast. However, this process of capacitation will take about 10 hours. Now, capacitation are changes, or is the word for all of the changes that have to happen in the head of the sperm so that the sperm will recognize an egg when it bumps into it, and so that when it does recognize it, it will release the digestive enzymes that will tunnel through the zona pellucida that will allow it to actually touch its cell membrane to the egg cell membrane. And that capacitation happens mostly in the sperm that are like bumping their way around. Sorry, let me use a pen. They're kind of bumping their way around. Oops, I'm a little lost. Oops, I'm a little lost. All of that bumping their way around actually makes it so that it takes them a longer time, maybe 10 hours, to get all the way up here. But all that bumping around causes the head of the sperm to interact with all of the lining cells of the woman's reproductive tract. And it seems to be, let's imagine that there's like a layer of stuff that is keeping that cell membrane from being able to notice that it has touched the zona pellucida of the female reproductive tract. And bumping around inside of the woman's reproductive tract for a while is what allows capacitation. The sperm that kind of take their time getting up to the infundibulum, they will more efficiently capacitate. And then when their cell membrane touches here, the zona pellucida, that causes their cell membrane to release those digestive enzymes that are here in green, boom, and then they digest that little tunnel through the zona pellucida, and then they will end up doing a fertilization event. So a couple of things. One thing is if intercourse happened like 48 hours before ovulation, then the egg will get ovulated and it'll just be out there and it'll go, okay, I'm ready to find, and then boom, fertilization happens. Because 48 hours, those sperm are capacitated and ready to go, right? However, it could happen that the ejaculation happens maybe 20 hours after ovulation, in which case this egg only has 4 hours to live. And then the sperm arrive in 10 minutes, boom. And she's like, oh, I'm so glad you're there. I only have 4 hours to live. And then the sperm will go, but baby, you know it takes me 10 hours to capacitate. and then there won't be a fertilization event, right? So there's more than a 24-hour window where intercourse and ejaculation can cause fertilization, but it's not an infinite amount of time in humans. All righty. We're going to go. Okay, so here's pictures of what I was talking about. Fertilization in mammals. So this is a sperm that has capacitated, so its cell membrane goes right through remember all the posse cells that came along, goes right through there and touches the zona pellucida, and then it goes, I've reached the zona pellucida. I'm going to go ahead and release my digestive enzymes. So it does the acrosome reaction, releases digestive enzymes. Digestive enzymes create a tunnel, and then when his cell membrane fuses with her cell membrane, then we actually get a fertilization event. Now, that fertilization event only happens once, all right? Some people are under the misunderstanding that if two sperm get into an egg, you get twins. No. If two sperm get into an egg, you do not get a pregnancy, okay? Two sperm in one egg, not compatible with won't even become an embryo, right? It'll stop dividing very, very quickly. So how does the cell stop it from being fertilized by more than one sperm? It does kind of an action potential sort of thing. So as soon as one sperm does this, then there's this like electrical zone that gets set up, and then there's no more sperm that are going to enter into that particular egg. So how do twins happen if it's not that? Well, one way that twins happen is that instead of just having one ovary sending out one egg, maybe both ovaries, they each sent out an egg. When two ovaries each send out an egg, then each one can be fertilized and can create a twin. And those are the kind of twins that we call fraternal, which are non-identical. As a matter of fact, if you've got twins like that with two eggs, they can have two different fathers. If during that little window of opportunity, there was intercourse with two different males, then you can actually have twins that do not have the same father. That can happen. Identical twins we will talk about when we start talking about development, and we will do that at the beginning of our next lecture.